All right, ladies and gentlemen, so we know a few of the players in this discovery of DNA, the molecule of heredity. We know about Francis Crick and James Watson. We know about Rosalind Franklin. And basically, these are the three main players. And they're all trying to figure out the structure of DNA, and both have a different picture of the puzzle that the other needs. So individually, they weren't really getting anywhere. So there's an additional player in this game, this discovery that we haven't talked about yet. And we're not going to talk about him very much at all. But we know our original three are James Watson, Francis Crick. Maurice Wilkins is the man we have not spoken about and probably won't much further. And then Rosalind Franklin. So it was about 1953, and Watson, all right, our gentleman here, was actually shown a copy of Rosalind Franklin's X-ray pattern photograph, the photograph from the last screencast. And as soon as he saw it, because he and Crick were such DNA experts, right, understanding what it was, the molecule was made of, and trying to figure out for so long what it looked like, he saw right away how DNA was arranged. He saw the double helix pattern with the nucleotides oriented inward. And within weeks, Watson and Crick, after seeing Rosalind Franklin's photo, came up with the structure of DNA, and that structure inherently explained how DNA could carry information and how it could be copied. And the way the structure communicated that was the sequence of nitrogen bases, right? Remember, nitrogen base is that part of the nucleotide. So it's the sequence of those nitrogen bases in DNA that allows DNA to carry information. It's a code, if you will. Here's a real photograph of Watson and Crick working on a 3D model of DNA. So Watson and Crick would ultimately describe the DNA molecule as a double helix, and that's still what we call it today. And you can think of it as a spiraling staircase, or like just two strands wrapped around each other. So let's just put things into a timeline here. In 1953, Watson and Crick would solve the structure of DNA, thanks to Rosalind Franklin. In 1958, 5 years later, Rosalind Franklin would die of ovarian cancer at the age of 37, likely caused by her exposure to X-rays, which were less than understood to be carcinogenic at the time. And in 1962, 9 years after announcing their discovery of DNA and its structure and how it could carry genetic information, Watson and Crick got the Nobel Prize. Now, the Watson and Crick model of DNA built off of Rosalind Franklin's Rosalind Franklin's photograph, of which she would not get credit for many, many years because she was a woman in science, and women in science were often taken advantage of. Their model of DNA is as follows. It was a double helix that looked like a twisted ladder, like we see here. And we know that that's the correct description of DNA because that's the one we still use today. That's the one that allows us to understand how genes work, how DNA fits inside the nucleus of a cell. So this structure, and we've been able to visualize it further beyond Rosalind Franklin's X-ray crystallography photographs with modern techniques, and modern techniques also show that DNA looks like this twisted ladder, this double helix. Now the sides of the ladder, right, the arms out here on the outside, not the middle, not the parts going across the middle, we know that they're formed by alternating sugar-phosphate bonds, right? The sugar group of one nucleotide bonded to the phosphate group of another. So these are alternating sugar-phosphate bonds. And the rungs of the ladder, right, the parts across the middle here in the DNA molecule, we know that they're formed by nucleotides. The nitrogen base group of a nucleotide, right? The nitrogen bases, remember there were 4 different ones: adenine, thymine, guanine, cytosine in DNA. So those are what create the rungs across the middle of this twisted ladder. And the two strands, right, because these are 2 individual molecules of DNA that are bonded together by interactions between the nitrogen bases of nucleotides, they're held together by some of the weakest bonds in nature, and those are bonds we're familiar with, hydrogen bonds. I think I told you back in Unit 2 that millions of hydrogen bonds are quite stable and keep one of the most important molecules for life stable, and that is DNA. So hydrogen bonds exist right here, right down the middle, right in between the nucleotide nitrogen base, between the nitrogen base of one nucleotide and the nitrogen base of another nucleotide. So the hydrogen bond exists between those nitrogen bases. So these hydrogen bonds, they form only between certain base pairs. Only between certain base pairs. Adenine is always bonded to thymine, and guanine is always bonded to cytosine, as you can see I have displayed here for you with colors. This is also why Chargaff's rule from a few slides ago makes sense. That's why the amount of adenine in a DNA sample is always the same as the amount of thymine, because adenine always binds to thymine. That's why the amount of guanine is always equivalent to the amount of cytosine, because guanine binds to cytosine. And we call these base pairing rules. And again, don't forget that we do break the nitrogen bases into purines and pyrimidines, right? AG for purines, they have the bigger double ring structure. TC for pyrimidines, they have the smaller single ring structure. And we call this complementary base pairing, right? A is complementary to T, T complementary to A, C complementary to G, and G complementary to C. They bind together via hydrogen bonds. So let's take a closer look at the structure of DNA. Now this is a DNA molecule, right? No different from the one we just looked at on the last slide, except the last one was more of a cartoon, whereas this one is the molecular structure. So we have our nitrogen bases. This is a thymine bonded to an adenine via these dotted lines in the middle, and we use dotted lines to represent hydrogen bonds because they're weak. Remember that from Unit 2. Here is our phosphate group bonded to our sugar group, bonded to another phosphate, forming our alternating sugar-phosphate backbone of DNA, right? The arms of the ladder, the outside of the molecule. And then, of course, we have our hydrogen bonds between the nitrogen bases in the middle. And we see cytosine paired with guanine, guanine paired with cytosine, thymine paired with adenine, and adenine paired with thymine. So A is always with T and C is always with G. All right, folks, I will catch you in the next one.